Great. Super. Thanks everybody for joining. People keep joining along the way. I'm Tony Oro, one of the co-directors for the Maternal and Child Health Research Institute, and we're really excited about have today's MCHRI seminar with Katrine Svensson and Tom Robinson. I think this is one of the, we try to focus on cutting edge programs, and this one is really at the top of the list here. I hope you really appreciate this group of investigators that are really focusing on interventions for early obesity, one of the big problems in our society right now. And so we'll get to that in a second. But first, a couple announcements. I want to thank our MCHRI Education Committee, the group listed here. These are talented individuals that basically are putting together this program and also our yearly seminar series. If you're interested in joining, please reach out to any of us. We'd love to have you. I think really the goal is to have a diverse set of education committee members who really represent across the entire campus maternal and child health research. Next slide. Coming up next in April, on April 14th, we have our Biodesign Faculty Fellows seminar series where we basically highlight three of the many Biodesign Faculty Fellow projects that are funded through MCHRI. And we have a list here of very exciting projects. So please tune in for that. And then a very important immunology of autoimmunity and antigen presentation symposium to honor our beloved Betsy Mellons, really a rock star of immunology. And this is really a incredible number of individuals that are going to get together to honor her and also to honor the science that she really pioneered. So that's Thursday, May 1st, 8 to 3. It's a hybrid event, so just register either on the MCHRI website or through the QR code that's listed here. There's a Stanford Medicine Children's Health Research Community event, which we do through MCHRI on an sort of quarterly basis to really highlight the research support office offerings that the MCHRI allows. If you're a scientist and you want to know how to basically do clinical and translational research, please use the MCHRI Clinical Research Support Office. We have it for you. It makes your life easier and makes your science go faster. And then finally, we have our 8th annual symposium, Wednesday, October 22nd. It's really going to be focused on maternal and fetal medicine. And so this is a really exciting program looking at the earliest ways to basically assess and predict disease at birth so that we can then, from the maternal and fetal interaction, so we can then intervene earlier to basically prevent things like childhood obesity. And so today, just wanted to, wanted just to point out, to introduce Katrine and Tom, who will talk about interventions for obesity from pharmacological peptides to community programs. Katrine's a assistant professor of pathology. We were very excited to recruit her here when she came. And Tom Robinson, who's a now professor in child health, professor of pediatrics, and really talks about this combination of epidemiology, population health, and basic science insights, which really have led to this amazing proposal for this obesity center that has gotten a pretty good score. So take it away.
Yeah, sure. Let me share my screen.
And thanks so much for joining us. For those who have questions, we can just, at the end, we either put them in the chat and I can read them off, or we can, or just unmute and we can raise your hand and we can have a good discussion. So Katrine.
Great. Thank you so much, Tony, for the introduction. So I'm Katrine. I run a basic science lab focused on energy regulation. And today I'll share a recent discovery of an anti-obesity peptide, some possible implications of our work, and where we're going next. So as you know, childhood obesity is one of the most serious public health challenges of our time. Between 2017 and 2020, nearly 1 in 5 aged 2 to 19 were living with obesity. And as you know, this is not just a number, it reflects widespread, early onset risk for lifelong health consequences. And this burden just increases with age from 12% among 2 to 5-year-olds up to 22% once they're 12 to 19. And this trajectory is really alarming because over 75% of children that have obesity will go on to have severe obesity as adults, which increases their risk for type 2 diabetes, cardiovascular disease, and early mortality. So early intervention is not optional, it's really imperative. And we have all heard of Ozempic and Wegovy and how these drugs have revolutionized obesity treatment for adults and children, in some cases over 12 years of age. And these drugs are peptides called GLP-1 that act by suppressing appetite. So today I'll talk about one of our recent findings of peptides similar to those of GLP-1. This paper was published just this month and received a lot of media attention, talking about this being a naturally occurring molecule that will compete with Ozempic. It's clearly a stretch, but I'll talk about how we think this peptide works and how we came to find this peptide and other peptides that we have found in my lab. I'm happy to take questions anytime. So we started out by trying to understand how we can use the information learned from monogenic obesity, and can we use this to understand general obesity in both children and adults. And the most common example of monogenic obesity is caused by an MC4R deletion or mutation, leading to hyperphagia, overeating, and obesity. And MC4R, similar to many monogenic obesity genes, have strong genetic links through a class of enzymes called proconvertases, PCSK enzymes. And in particular, PCSK1 was one of the first monogenic obesity genes that were identified. And you can see here in the picture to the left, a child with a PCSK1 mutation with severe early onset obesity, weighing almost 80 pounds at 3 years old. And the reason why this mutation is leading to obesity is because if you're lacking PCSK1, you can no longer produce many of the peptide hormones that we have that regulate appetite, including alpha MSH, which is the ligand, peptide ligand for MC4R. You can no longer produce GLP-1, which acts on the GLP-1 receptor and controls feeding. So this obesity phenotype is also conserved down to mice. So if we delete or mutate PCSK1 in mice, they develop a rapid and massive obesity due to overfeeding. They just cannot stop eating. So the way that this enzyme works is to first cleave a precursor protein into a smaller peptide, which then act on the receptor. So for example, for MC4R, the precursor gene is POMC. It will be cleaved by PCSK1 into alpha MSH, and then alpha MSH will act on the MC4R receptor. In the case of GLP-1, the precursor gene is proglucagon that can be cleaved into multiple peptides by PCSK1, including GLP-1, that act on GLP-1 receptor. So if you look at the entire system, mutations in either the processing enzymes, PCSK1, some modifying enzymes called CPE and PAM, or in the receptors that are responding to these peptides, any mutation in humans lead to either obesity or an endocrine defect. So this entire system of peptide processing appears to be one of the most relevant systems that regulates body weight in humans, and especially PCSK1, which I'll focus on today. So for many years, people have tried to identify new peptides, peptides similar to GLP-1, but they're extremely low abundant. very difficult to find using mass spectrometry. You only find the most high abundant proteins like plasma proteins, adiponectins, sometimes interleukins, but you don't find insulin, GLP-1 with regular mass spectrometry. So we wanted to know, can we leverage what we know about the PCSK1 biology to identify new peptides and identify those that have gone undetected by traditional approaches? So what we did, instead of going in with mass spectrometry, we generated a computational algorithm to predict new peptide hormones based on the cleavage site that are being recognized by PCSK1. So in our genome, we have 2,000 secreted human proteins. And what we did was to annotate those cleavage sites that are being recognized by PCSK1. So these are dibasic cleavage sites at a certain frequency in a secreted protein. And in doing so, we predict that there are around 370 prohormones instead of, the 50 that we know of. And that is predicted to generate around 2,600 peptides across all human tissues. So this is an analysis that surprisingly nobody had done before. And we find many more peptides than anyone would have anticipated. So we're looking for things that are similar to a pre-proglucagon or POMC in terms of their length, sequence recognition, and the number of peptides that is being generated. So we're focusing on the things that look like a prohormone pretty much. And you can ask, well, how accurate is this prediction platform? And if we take proglucagon as an example, on the top, I'm showing you in gray the known peptide fragments that have been experimentally validated from the proglucagon gene, including glucagon, oxyntomodulin, GLP-1, GLP-2. And on the bottom, I'm showing you the peptide predictor and the 6 peptides that we are predicting to come out from this method. So if you wouldn't have known about GLP-1, you would have found GLP-1 as proglucagon peptide number 5. So we can now do this across the any tissue data set. So we have done this across the entire human tissue proteome and ask, where are these peptides coming from? So in this graph, I'm showing you tissues where these peptides are expressed and released from. So from brain, we find 400 peptides, from the liver, 300 peptides, and then a small number from our classical endocrine organs. And we have been focusing a lot on the brain because these neuropeptides looked very similar to in sort of the pattern of cleavage prediction as common neuropeptides that we know of. So if you compare to what we know, here I'm showing you a graph of known peptides in purple and unknown in gray. And by known, I mean a peptide that has a known GPCR and known activity or function. And you can easily appreciate that we have covered here a very large biological space of peptides that people have not looked at before, especially those that are smaller in length. They seem to be more potent in terms of bioactivity. So to make a very long story short, we screened a number of these peptides and identified a 12 amino acid peptide that we call BRP. It's cleaved from a larger precursor protein called BRIMP2, which is only expressed in the brain. It's released, it's an internal fragment that is being released and cleaved by PCSK1. And we can confirm this by actually detecting this exact peptide sequence in mice, and we can look at it in wild-type mice, or we can look at it in PCSK mutant mice that are lacking this enzyme for cleavage, and we actually don't detect it in the mice that have this cleavage defect. So what does this peptide actually do? So it turns out that it's elevated after a meal. So if you have mice that are being fasted overnight, and then you refeed them either a regular diet, low-fat diet, or a high-fat diet, the levels go up after feeding. So we detect the peptide levels in the brain at 0.4 nanomolar normally, and then it goes up after feeding, acutely after feeding, 30 minutes after feeding. So this would indicate to us that this peptide is responding to either glucose or anything else in the meal to regulate feeding. So to test this directly, we used metabolic cages, which is in one of the core facilities, a part of the STRC in NORC, where we can measure all sorts of metabolic parameters, including food intake. So here we synthesize the peptide, which is shown in purple, and then we give the mice a single injection of this peptide. Here at a dose of 5 milligrams per kilogram, which is not a crazy high dose. It's a pharmacological dose, but it's not insanely high. We give the mice a single injection and then measure food intake, and you can see that there's a suppression in food intake up to 16 hours. And importantly, if you take the same sequence, 12 amino acids, but you scramble the order of the sequence, it no longer has this effect. So it's really important that it is in that particular order, suggesting that it's a specific effect. And what made us really excited about this, the effect of this peptide, is that if you take an unmodified GLP-1, so not liraglutide, semaglutide, but just a non-modified GLP-1, do the same experiment there, we see the same type of food intake suppression in terms of magnitude. So this told us that this looks pretty similar to a drug that we already have working in humans. So many things can lower appetite due to effects that are not desirable, including toxicity, nausea, other types of sickness responses. So to rule out some of these effects, we actually performed 12 different experiments in 3 different animal species, mice, rats, and pigs, to look at different behavioral effects. And I'm showing you some of them here. So in the top, I'm showing you movement after peptide injection, which is not affected. We tested movement and coordination using a Rotarod test in mice. And we also looked at forced swim test after peptide injection, which is not different or affected. One of the major side effects of GLP-1 based drugs is delayed gastric emptying and constipation. So we wanted to look at that as well. So in the bottom left graph, I'm showing you gastric emptying test, where we look at how fast the stomach is emptied. And we see no effect of BRP there, but we do see a suppression with Exendin 4, which is a GLP-1 receptor agonist. Other assays looking at behavior include fecal pellets, water intake, open field assays, and anxiety test. And then we also looked at sucrose preference. And in none of these tests, we saw any adverse effects. Another test that also it would test positive for GLP-1 receptor agonists is a test called conditioned taste aversion. So this is a test in mice that basically measures how nauseous or sick they feel after injection. And as you know, many patients that are taking GLP-1 receptor agonists feel dose-dependent nausea, and some even vomiting. Mice cannot vomit, so this is sort of the best test that we have to look at this in mice. But basically, the way that this test works is that you inject an agent, in this case either BRP or our positive control lithium chloride, which is a nauseating agent. You can also use a chemotherapy that will cause nausea. And you do that at the same time as you offering the mice to drink saccharine water. So if you do this repeatedly over a couple of days, they will associate this saccharine water with nausea. And on the last day, you don't inject them with anything and then just look at what do they prefer, water or saccharin water. So if you do this with vehicle, which is just a saline solution, mice will always prefer saccharin. We see the same thing with BRP. It doesn't affect their preference for saccharin, which would indicate that it doesn't induce any nausea or aversive response. But for example, lithium chloride, heavily nauseating agent, will make the mice develop aversion towards saccharin water. So as you know, mice are very different than humans, and they have a different eating pattern. They eat at night, and they don't eat the meal sizes that we do. So to really assess the translational relevance of our findings in the mice, we performed similar studies in Yucatan mini pigs. This was actually done downstairs in the animal facility here at Stanford. We bought 3-month-old Yucatan mini pigs, gave them a single intramuscular injection of BRP, the human BRP, and then looked at food intake over 3 hours. We also did a tail scoring test, which is a test that tests for anxiety or stress. And what we saw was very similar to what we saw in mice. This is a dose that is adjusted to the size of the pig and also converted. So basically, it's a very similar dose to what a human would get. Here we see a 50% suppression of food intake in Yucatan mini pigs. And we can actually do experiments where we give them either a vehicle one day or BRP the next day. And then the third day, because the half-life is very short, they will go back and receive a vehicle again, and the food intake is restored. And on the last day, we injected liraglutide, which is a modified long-acting version of GLP-1. And again, the effect size is very similar. So this was very encouraging to us that this is an effect that is translatable from mice to pigs. And it looks identical to those drugs that are being used in humans. So if you now go in obese mice, so these are mice that have been on a high-fat diet for 6 weeks, so they are obese. We give them a continuous repeated daily injection of the peptide compared to liraglutide. We actually see a dose-dependent weight loss. I'm only showing you one dose here, but we see a dose-dependent weight loss over 14 days. And after 14 days, they have lost 10 to 15% of their body weight. And they improved their diabetes as well. And the magnitude of effect is similar to that of liraglutide. If we were to compare it to semaglutide, it would be performing worse. So, but the effect is very strong for being a completely unmodified, short-lived peptide. So to understand really how it works mechanistically, we looked in 3 different models of mice. Here I'm showing you leptin-deficient mice, MC4R deficient mice, and mice that have been treated with a GLP-1 receptor inhibitor. And this was to see if we block the pathways that we know are responsible for feeding regulation in mice, leptin is well known, MC4R is well known, then that may tell us what pathways are involved. But what happened was that we don't see that it's dependent on any of these known pathways. The peptide suppresses food intake under all these conditions, even if you delete leptin, MC4R, or if you don't have GLP-1 receptor active. So it's a completely different pathway. So we started to collaborate with Li-Chun Liu, a neuroscientist here at Stanford, and did whole brain fast mapping to see what areas of the brain are actually activated by BRP, and are these areas any of the known areas that people haven't looked at before. So across the entire brain, we see activity. So basically the experiment is we inject the peptide. After 1 hour, we take out the brain and stain for CFOS as a marker of bioactivity. This is a standard test. And we see basically 4 different brain regions that are lighting up. These are all brain regions in the hypothalamus. So VMH, DMH, arcuate nucleus, that have previously been associated with feeding, but they're not the classical POMC neurons that people have looked at before. And it's not the same neurons that are activated by GLP-1. So we're currently looking at what is exactly the neurons that are needed for this activity. Can we delete them and lose the effect? So many people ask me, how is BRP actually different from GLP-1? And GLP-1 acts throughout the body, not just in the brain to suppress feeding, but also in the GI tract to lower gastric emptying and slow down transit time, causing constipation. It acts on the pancreas to increase insulin secretion. That's why it was a diabetes drug. And it also lowers adiposity and lowers lean mass as well as a consequence. And what we see is basically none of the peripheral effects. Even if you administer BRP peripherally, we only see activity in the brain. We don't see anything in the GI tract, in the pancreas. We only see weight loss mediated by food intake suppression. So what we're doing next is to understand, well, how can we use this, or can we use this in terms of anti-obesity treatment? For who would it be most beneficial? Can we understand more about the mechanism by finding the receptor? What does this peptide do endogenously? We don't know much about what are the levels, how is it changing? Does this peptide have any physiological relevance? And we're also working on finding other similar peptides, which we already have, but they work differently. So, but there are many things coming out from the peptide prediction platform. So with that, I want to thank my lab, entire lab, working so hard on all these projects, our collaborators. We have worked a lot with Danny Xu on trying to optimize these peptides, understand the chemistry of them. So that has been a great collaboration in our funding from NIH and industry. So thank you so much, and I'm happy to take any questions.
So maybe we'll go on with Tom's presentation, and we'll take questions at the end. Thanks so much, Katrina. That was an amazing presentation, and congratulations on your recent paper that came out. It's just a beautiful piece of work. Tom, do you want to take over?
Whoops. Sure. Thanks, Katrine. That's and I'm gonna sort of change the frame to more of things we're doing to try and develop better treatments and preventive interventions outside the body by focusing on mainly on the community setting. And I wanted to preface it also by saying recently I was at a meeting where I had a longtime colleague say to me that congratulate me that saying Stanford's now become the place to do community-based childhood obesity research. And because now I'm only one of a whole bunch of different people in our department doing clinical community population policy research in around obesity in children. I think in the past month or so, you've heard from Anna Grumman and Jennifer Wu Bidell, two fairly recent additions to our department about their really exciting research in this area. Many of you also are familiar with Anisha Patel's research and Lee Sanders' clinic-based studies. Just recently got a lot of attention for great outcomes in a clinic-based study done here. We also have very robust clinical programs here at Stanford, the Pediatric Weight Clinic, which is a more standard subspecialty clinic for children with obesity and particularly severe obesity, a very active adolescent bariatric surgery program, and our Stanford Children's Health Pediatric Weight Control Program, which is really one of only about a dozen programs in the whole country that follows or implements a family-based behavioral approach that is the only approach currently recommended by the U.S. Preventive Services Task Force. And of course, in addition to that, all of you in every subspecialty, in every aspect of our department across are dealing with kids. Obesity has become, because it's 1 in 5 kids now, it's become a part of everybody's care, no matter what subspecialty you're part of. So I'm going to focus specifically on a couple areas. I don't have time to cover everything we're doing, but a couple areas that I think are interesting and give you an idea about themes that run through a lot of our research, and that's stealth interventions and our new research in screenomics. To start off with, let's see. So anytime you, to try and develop a strategy for disease prevention and control, there are many ways you could come about it. You could try and change the environment. You can try and introduce policies. You can try and do something interpersonal interventions, or you can work, or individuals can try and change them themselves, such as self-regulatory behaviors. But the common final pathway of all of those is changing behavior. And so that's where we've really focused. So even if you have a great new drug that is gonna solve the obesity issue, you still need to get people to take it. And so as we've seen recently, for example, with vaccines, they're really, really good, but only if they're used. So our research is founded in Bandura's social cognitive theory. I find it the most most useful as an intervention designer. And there are 4 key learning processes in social cognitive theory that come through in terms of how to help people change behavior. One is attentional processes, and that is you need to attend to the intervention itself. It needs to be aware of it and seen. Retention or retentional processes, which is they need to be remembered, and it needs to be something that is people are able to remember what they're supposed to do. Production processes, which is turning those instructions or those memories into action, and is highly linked with perceived self-efficacy, which is a key component of social cognitive theory, and motivational processes. Those are the things that get you sort of across the line. How do you initially adopt a program and stick with it? I focused mainly in our group, we focus mainly on motivational processes, because those are the things that have been really, I think, neglected a lot in prior research. To give you and I divide motivation into 2 categories. One I call outcome motivations to adopt the new behavior, generally the outcomes of that. What are you going to get from adopting a new behavior? But also what I call process motivation, which is motivation to participate in the intervention itself. So not just what you're going to get from it by adopting the behavior, but is adoption of the behavior itself motivating? That's the area that I think has been ignored the most in most research, behavior change research. So that's where we put a lot of our attention. To give you examples of these, outcome motivations are things like obesity and diabetes and high blood pressure, health things where we try and convince people to change their behaviors because it's good for them. Whereas if you look at the last 70 years or so of social psychology research, you see a whole area of what are called intrinsic motivators, things that are really what drive behavior change, not necessarily the knowledge of what the outcomes are, but what this part is the sort of the process motivators that keep you engaged in an intervention and help you maintain that behavior over time. So those are things like perceptions of choice and control, that you have some choice and control over what you're doing, levels of curiosity, goals and challenge, fantasy and contextualization of content. And that's, it's used a lot in what now has sort of been popularized as gamification, perceived individualization, that you feel that the intervention is specific to you. Both cooperation and competition are both highly intrinsically motivating. Social interaction feedback is highly motivating. Pride, sense of accomplishment are motivators. And for kids, peer and parent approval are quite important motivators, intrinsic motivators, to keep people engaged in whatever your goal is for them, for their behavior. So when I look at that list, I see nothing about health on the right side. So I came up with the question, well, does a health behavior change intervention need to look, feel, sound, smell, or taste like health education? In fact, do we need to discuss health at all to change behavior that will improve health? And that's where the idea of stealth interventions came from, not because they are deceptive in any way, they're not stealthy in that way, but they're stealthy in that the targeted behaviors, and in many of these for obesity, it's physical activity or reduced inactivity or changes in diet, end up being side effects of the intervention from the point of view of the person participating in it. So they're doing it to meet their motivational needs, to be able to do something because they would do it anyway, because it's motivating to them. And I get the benefit of saying, well, it also improves their health as well. So let me give you some examples of that. Some of our early work was on screen time, and this work was done before we even had the screens I'm going to talk about later, before we had phones, but looking at television viewing. And there were a number of studies that showed that television viewing was associated with weight gain and obesity in kids and amount of television viewing. And so we decided, let's see if we can develop interventions to reduce screen time, reduce television viewing, which some people said, well, that will be impossible. It's too addictive. It's too difficult for kids to do that. But we took those same principles. We didn't do anything different about making them want to see necessarily what the positive outcomes were going to be, although it wasn't about health or about certainly not about obesity for this. It was about perceived choice and control and perceived individualization and cooperation and competition, all those factors. And in doing that, we built interventions for schools and then for individual families that helped families and help kids reduce their screen time. What we saw, and when we did randomized controlled trials of that, is that we saw, in fact, we were able to reduce screen time in kids in those controlled trials. And with that, we got less weight gain without ever mentioning obesity, without ever mentioning nutrition or physical activity, less weight gain associated with that, less aggressive behavior when we did objective measures of kids being aggressive to each other on the playground, for example, and less consumerism, less requests for parents to buy them stuff they saw advertised, and as well as that, improved test scores, just from interventions to reduce screen time that were designed based upon those same motivational principles. Other examples, what about getting kids to become more physically active? Well, one of the challenges in kids, especially kids with obesity, is to get them out and active. And in particular, they don't sort of join many sports teams, which many of us in our lives have found to be highly motivating and kept us going. And so this was actually Dana Weintraub's brainchild to start. Could we start a team sports league specifically just for overweight kids, where they weren't worried about being necessarily the last one picked or the slowest one on the field, because all the kids would be similar to them, and their perception was that would be less threatening. At least that's what we found in focus groups with them. And so we tried it, and in fact, it worked really well as they showed up, and attendance rates were really high. And again, we didn't, it just wasn't your regular sports team. It was, in contrast to that, we call it team sports, which is different. So how do you take all the positive motivational aspects of team sports without all the necessarily highly competitive and sometimes very difficult things for some kids of sports teams, or the highly competitive sports teams that are out there, where it looks the same to them and it looks the same to anyone watching. It's designed very differently with those things like perceived choice and control and things, and where do you build in things to make it highly motivating? Well, things like the uniforms they have, and the team name, and the shin guards, and having young adult coaches who are highly motivated. All those are really great things, and that's why the kids participate, not because they're losing weight, not because they're getting more fit. It's because of all those other aspects that are intrinsically motivating, that process motivation that keeps them going. And when we tested this in randomized controlled trials, where we were comparing it to health education, so what you would get if you were going to regular classes about nutrition and living a healthy lifestyle, is that kids did become more physically active. than the kids in the controls, and they lost BMI compared to kids in controls over just over 6 weeks in our initial trials, 6 to 12 weeks. Another one of those is dance programs that we've done for girls. A lot of this came out of just the observation that when music is playing, many girls just start moving naturally. Many boys do too, until they notice you're watching them, and then they stop. But it's one of those things as humans where we tend to music and rhythm is sort of natural to us. So we said, how can we do this in a way where we apply those same principles? These are pictures from a project we did with Mexican-American girls and Ballet Folklorico, where we're teaching them with tradition and folk dances from Mexico, and with the amazing costumes, and they make their earrings and the choreography, and again, having young adult dance leaders or dance teachers who they can look up to. And when you look at the smiles on these girls' faces, performance is also highly motivating to their parents and family. You see that just how much fun they're really having when they are participating in this. Again, it's not about health, it's not about weight, it's not about fitness, it's about dance, and it's about your culture and learning about something culturally relevant to your family. We did the same type of trial in African-American girls in Oakland, and low-income African-American girls, where we used hip hop, step, and traditional African dance. And again, highly successful. In one of these studies with 2-year outcomes, we saw, as a result of these dance interventions, lower cholesterol levels, significantly less prediabetes. We actually cut the prediabetes rate by 2/3 compared to a control group that was getting health education, again, health fairs and information about how to improve their health. Greater cultural identity, so positive, and also less depressive symptoms in girls just from being part of this compared to controls. So with that, I also started thinking, well, what is even more motivating than some of these other activities that we're doing, where people join and sustain them for very long periods of time? And then, and really the classic is sort of religious movements, but social and ideological movements in which people do things and stick with them, even despite a lot of pressures in the environment to not participate in them. And so there's a whole list that I came up with back when I thought of these ideas back 15 years ago. Wow, that's a long time ago. But 15 years ago when we first published this, which was of examples of types of social movements that overlapped in their behaviors and their goals with our goals for obesity prevention and treatment. One of the, and so our first test of this was a trial I did with Christopher Gardner, where we designed a class that we ended up teaching for 10 years, or more than 10 years, in human biology, called Food and Society, where we taught a class that was all focused on the non-health, non-nutrition aspects of food and agriculture. So sustainability issues and ethical issues and labor issues and global trade issues. And in doing that, we couldn't randomize Stanford students to take our class, but we could look and compare students who took our class to students who took classes on obesity, who took classes on community nutrition, public health nutrition, things like that. And with that, compare them from beginning to end at changes in their behaviors, their dietary behaviors. And what we found is, even though they all started out very similar at the beginning of the quarter, by the end of the quarter, the students who took our classes had changed their diet significantly more than in a positive way than students who had taken courses about nutrition, even though our course wasn't about nutrition. It was about all these other aspects of food. Similarly, we've done a couple randomized trials of environmental sustainability interventions. This is a picture from one that we did where we randomized 30 Girl Scout troops in the local area, and half of them did. This was actually a study funded by the Department of Energy, U.S. Department of Energy, where we randomized half of these troops to learn about reducing their household energy, and the other half to reducing energy from food and transportation sources by choosing different foods of what to eat. And that mango sort of doesn't really count there because that has to be flown a long way. But in general, fruits and vegetables have a much lower carbon footprint and much less electricity or energy use than livestock, for example. And so having those two as control groups for each other, we saw that the interventions worked at the one focused on food and transportation at increasing activity levels and improving diet compared to the control focusing on household energy with that. So we've now sort of shown this in several studies that this is an appropriate or a successful approach to take. Let's see, I have to mention too that, of course, as Girl Scouts, another intrinsic or another extrinsic, I guess, motivator for Girl Scouts are patches. So we even had our own patches. Now, why do I think that these types of interventions work? Well, they focus on your identity, your values, your beliefs, your emotions, things that are below the surface, not those things that are obvious, sort of what are you going to gain from this, but the things that are below the surface. Social interaction, membership in a group, having models as a part of a group who model the behavior you want them to adopt, very important in behavior change. Social support for behavior change, perceived social belonging, and perceptions of collective efficacy, that as a group you can accomplish something more than you could as a individual. Less risk of failure as well as part of a group. If you're trying to end climate change, it's less, and you don't succeed, you don't feel like a personal failure. If you step on the scale and you haven't lost weight, or you don't make it to your after-school sports program, that is a form of failure that you feel that harms your self-efficacy, as opposed to doing something that you are doing and just participating is success itself. And also, if these are successful, they change norms and policies which reinforce the behaviors you're trying to change. Okay, I will quickly jump to screenomics and the Human Screenome Project, which I'm doing with collaborators in communication psychology in the Graduate School of Education. And this is a project focused on a different environment and changing behavior through changing what you get through your screens. And now much of our lives are lived through our screens, especially the smartphone screens that we carry with us everywhere we go. And so as a result of this, we have the chance to try and understand what's going on people's screens and intervene in that context. This is an example of this software we created that we can put on your smartphone, and it takes a picture of the screen every 5 seconds. And so with this, we become like the only people, at least, will admit to being able to see everything people see and do on their screens. So this is an example of just a few hours of a student, their screen, and they volunteered to do this and have us show this, that shows you what people's screen lives are like. They're going all over the place. It's not the way we used to consume media, not the way we consume the rest of our lives. That timeline, that sort of barcode on the bottom, is categories of screens and also different valence and emotional content on the screen that we can actually do and code for each screen. So what do we see in the screenome and what have we learned from it? Well, one, there's tremendous idiosyncrasy across individuals and within individuals. There's huge between person and within person variance that we see, in that no two people's screenome look anything alike, and no 2 hours of your day look anything alike on your screenome. Interdependence of traditional content categories, in that there's a blurring. Our life is not divided into health, politics, relationships, shopping, finance. It's all blurred together. It's all together now, and it's the way we experience it on our screens. Also, our life is divided into short experiences, 5 to 10 seconds between switches, with the idea that there are possibly very consequential, very brief exposures that we're getting that have outsized effects on our outcomes, whether it's mental health, or food or anything. And that I'm bringing up food because that's how it applies to obesity here. We're studying food. Fragmentation of experiences, that you are actually, you and the algorithms are personalizing the threading of different content all together in different orders. And sequences and rhythms of use, in that you have to think in multiple time domains. Is how does this, it's all connected, but how do you relate the exposure you had 5 seconds ago to the one you have 5 hours ago and 5 days ago and a year ago, and how those all come together to sort of predict outcomes and change behavior. So we do a lot of work on trying to, it turns out it's a lot harder than it sounded when we first started, that AI models are not as well suited to this as we had hoped they'd be, because these are much more complex images, much more complex data than most of them are trained upon, which is pretty simple. And so we're able to take and identify textual features on all the screen through OCR. We identify what the graphical features are, either sort of embeddings or actual labels of things. And we combine that with a lot of human labeling to try and validate our models to see what's actually on the screens and what's being done. Here's an example from food of one 13-year-old screen where 37% of all of her screens in a day had some food content on it, which is really remarkable. And it wasn't just in social media or food videos. It was everywhere. It was in games. And this girl in particular had spent a lot of time playing games around food. So what is our goals with this? In my last couple minutes is first understanding digital media uses. How do people use their phones and use media? We don't really know at this point. The last 70 years or so of 50, 70 years of media research has all been based on self-reports of what people say that they saw in media. And so can we describe it in terms of the content, the function, the context, the time, and the interdependence of different stimuli? Precision diagnostics and prognostics, what I'm calling SWAS, or screenome-wide association studies. Are there ways that we can link screnotypes to phenotypes, both between individuals, like what is mostly done in biology, where you're saying, your genes are different than my genes, but also within individuals, where we're saying, what about, you an hour ago versus you now? How does that change? And how do those within person screnotype changes, how does that, or those screnotypes, does that influence behavior? The ability to sort of look at what are often called natural experiments, where you see exposures in the screen and see what comes out from them that may be very brief, for example, and the ability to redefine disease, health, and well-being, in that almost all of our definitions of what makes you sick or what makes you healthy or what makes you are really based on, are limited by the measures we use to assess those things. And so by looking at things every 5 seconds, and we can actually go down to every 1 second, but every 5 seconds, it really changes your perspective on how you would redefine something. How would you redefine depression, for example, or eating, for example, if you're seeing food spread out and eating spread out throughout the entire day as opposed to in meals, the way it's often measured. So, and finally, as it gets to prevention and treatment, can we develop truly personalized precision interventions? So real-time, closed loop, within-person precision interventions, the right intervention to the right person or group, in the right way, in the right dose, in the right combination, at the right time and place, to be able to maximize the benefits and minimize harms and waste. And in doing so, also hopefully reduce risk for bias and discrimination, where we can really precisely intervene in someone's screenome to change their behavior, and then test that in a way iteratively, just like they do to sell you stuff on the internet. Okay, so that was my quick run through several of the areas we're studying.
Thanks so much, Tom. I think just a mind-blowing session today, showing the mechanistic insights of critical endocrinology, mind-body interaction that Katrina talked about with BRP, and also this environmental group. This really shows why Stanford's child obesity program is so impactful and will be in the future. So thanks very much for both of you. Please, if you have any questions, we have a few minutes for questions. Maybe I'll ask the first one to Katrine. So it looks like BRIN-P2 is actually a brain, hypothalamic, and also pituitary derived peptide. Is that where you think it's secreted from mainly in terms of, and do we know what regulates its levels? Is it coincident with BMP2 or is it actually, are they separate?
Yeah, so the parent protein, BRMP2, is expressed in the cortex, hypothalamus, and it's the peptide or the whole secreted protein is not detected in plasma. So I think it's local, local secretion, but we haven't seen any gene expression changes of the parent protein. So after fasting and feeding, there's no change. So it's only changes in the peptide levels, not the entire parent protein, but we haven't done extensive analysis on that.
And is the enzyme that cleaves it, is it activity also regulated by feeding, or is it, or by cycling MP?
The enzyme that is cleaving it, PCSK1, is not regulated by anything. It's pretty highly expressed across the entire brain. It's known to cleave many neuropeptides, but it's not itself regulated.
Okay. Other quick questions. I think Elizabeth has a question. As a physician in the pediatric weight clinic, I've seen amazing behavioral changes in patients who are losing weight on GLP-1. As they lose weight, kids are more motivated to be active and healthier. This is a ripe area for research, and wonder about your thoughts about how to transition kids off of GLP-1 agonists while maintaining their behavioral changes, since most patients who start these meds do not think they'll be on them for long term. Maybe Tom?
Yeah, I think Elizabeth knows this is a hot area of discussion at this point, because it's not many people actually don't, even when they start them, even if they have success with them, don't stay on them very long, and for a lot of different reasons, and so yeah, as you know, I think, Elizabeth, I think that the jury's out on what the best ways are, but there are a lot of us trying to coordinate and identify strategies that will help. In about a couple decades ago, there was a bunch of research where they had less effective drugs, and they tried to decide whether you should start behavioral programs before adding the drugs, or do them at the same time, or do them after. And in those cases, none of those strategies really worked. It turns out that drugs were so much more effective that that people didn't really learn the behavioral strategies. And some of those were done here at Stanford by Stuart Agris and groups. But it's so there's a lot to learn about how we're going to be able to use all these. But I think there's always going to be a need for behavioral treatments, because clearly it's only a minority of people who are on the drugs. If Katrine can come up with a drug that doesn't have any side effects, that'll help a lot, especially if it's affordable. But it's at this point, the majority of children, the majority of adults with obesity are still sort of left with behavioral treatments at this point.
Can I ask as a follow-up, which is, the brain can adapt as well. So in theory, there might be peptides that can actually affect the set point brain, on a longer term basis in terms of, potentiation, long-term potentiation in a brain sort of way. Katrina, do you have any thoughts about, are any of your peptides, FSTL1, all of the beautiful ones you identified in your paper, any of those have any of those long-term?
Yeah, so we have not seen, they all look like GLP-1 type of mechanisms. So as soon as you stop them, you lose all the effects, and there's no memory or anything. You just basically go back to your old habits. So that is sort of a question I have for Tom also. What is it is that a risk, and what are sort of long-term consequences of going on these drugs without changing any of the behaviors? Or you just need to eat less. You don't even need to eat healthy, and you will lose weight on these drugs. But I wish there was something that would chronically change your satiety level, but so far we haven't found any example of that, and I don't know if that's possible.
Yeah, and that is one of the things that many of us worry about is there are benefits to physical activity and changes in diet beyond just weight,
Yes.
losing weight. And so in many people on these drugs, unless they're also changing those behaviors, they're not getting those other benefits.
Okay. Another question for Robinson: are there any budding ideas for stealth interventions related to the ongoing screenomics work?
Yeah, I mean, the idea, well, yeah, is using the same principles and doing there where we alter people's screenomes, where we can do that. You can actually, through even now with parental controls stuff, there are ways that people are already developing software to 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 do sort of more precision interventions with that. For example, if you have an interest in changing behavior around something, or a parent with an interest in changing your child's behavior, we're getting to the point where the technology allows us to identify what's on the screen on the phone itself. It doesn't have to be sent to us, securely, and for us to come up with all these machine learning models to decipher it, where that can happen right on the phone itself and can intervene right then and there. We already can do that with specific apps. So if a specific app comes up, that's easy to identify and intervene at that point. So, for example, there's laws about warning labels where there's legislation that's pending about warning labels for social media that could be detected right there in the operating system right there, and it would never have to leave the phone. So yeah, we would hope to do similar types of stealth interventions and apply the motivation. So it wouldn't just be stop using social media because it's bad for your mental health, which is a traditional way of regulation trying to change behavior. But it would be focusing on what are the goals and the identity, the things that the user themselves identify with, which you could get from the screenome, things that they're interested in.
Awesome.
So that's exactly what we're thinking.
Other burning questions? Yeah, I guess that's also called deleting the app. Anyway, if not, just want to thank Katrina and Tom for, again, it's absolutely scintillating hour from both sides of the translational spectrum. I really appreciate the effort, and good luck with the program getting funded. And we look forward to more and better results and interventions in the future. So thanks so much, and have a great day, everybody.
Thank you.